is the actual meaning of our biotechnology, or what is the meaning of chemical aspect of biotechnology, how we can use them, right? So till now we have studied that biotechnology is basically the application of principles of life sciences in biology or in our daily life, right? But the use of chemical engineering, or with the help of chemical principles, we can modify them and we can make it much more advantageous for our lives. Okay? Till now, we have studied that what are the benefits we can obtain through biotechnology in agricultural field, in food quality field, etc. Right? And we can develop certain kind of food or certain kind of plants which are disease protecting plants. We can develop certain kind of crops which are actually very beneficial for both the farmers as well as for the consumers also. Right? It is cost effective, it will reduce the production cost, it will reduce the transportation cost and storage cost also. So in this manner, it proves very beneficial in agriculture as well as it is already very beneficial for the food quality field. Right? We can produce the food, we can improve the quality of the food through biotechnology or through application of the combination of chemical field and biotechnological field. This is in general can be known as biochemical field. Right? This is a branch with which we can get much more benefits. Okay. Now, in this particular session, we will begin with benefits through or applications of biotechnology in medicines. Till now, we have seen it applications in the agriculture field and in the food quality. Now, we will see its application in medicine field. Here, we know, or in current scenario, we all know that India is lacking certain kind of medical facilities, right? To improve such kind of condition, this can be helpful. How? Let's see it. We have developed one technology which is known as rDNA technology. It is known as recombinant DNA technology. Okay. This biotechnological application is very important in healthcare sector because it allows for the mass production of safe and more effective medicines. Okay, that means I should say that here we cannot produce our vaccines or our medicines in a mass production manner. If we know this technology, we apply this technology on a large scale, then we can produce it on a large amount also. Okay, we will see that what is this recombinant DNA technology. Okay, so first is genetically engineered insulin. Till now, whatever we have seen that in diabetic patients, insulin is produced less by pancreas, and because of that, the sugar will not get metabolized. And if it doesn't get metabolized, then will run in our blood vessels. Because of that, it creates a problem. It creates diabetes. Right? To reduce that, we have to use insulin. Now, if this insulin is genetically engineered and it is active in nature, then at a certain time or after a certain period, it will give rise to a better condition of a patient. That means it is possible that the diabetes mellitus can be lowered and even can be treated also. Right? This is the thing that we can use in our medicine. Another one is gene therapy. Gene therapy is basically a biotechnological application involving a collection of matters that can correct a gene defect in a child or an embryo. Right? It involves inserting a normal gene into the possum cells or tissues to compensate the non-functional gene. Okay. Now, we all have studied that we all have genetically manufactured, or we all know that certain genes are responsible for disease, right? Like certain genes are responsible which came from our parents for us, because diabetes is an hereditary disease. If you don't follow a proper lifestyle, then you cannot get it. Similar way, thyroid or any other kind of genetically or genetic defect, which is known as genetic disease, right? That if it is diagnosed at an embryo level only, then we can treat that particular disease at that embryo level. That gene is removed and a proper improved quality genetic sequence is inserted into that particular embryo, and the child will be healthy. A person or a healthy person can lead a life easily. Through this gene therapy, we can reduce many things. Cancer can be treated, diabetes can be treated, certain hereditary diseases can be treated, even obesity can be treated, because certain genes are responsible for obesity. And when we use them, or when we treat them with the use of gene therapy, this problem can be totally removed. Right? It is possible, and it is actually implemented in certain countries also. Right? But it requires more research and development. Okay? Then we all know that we have a molecular diagnosis. So that is again a thing. We have to develop, or we have to generate a particular gene sequence. We will generate it. We will diagnose that particular thing, and we can remove that particular gene and treat a molecular disease. Right? It is possible for all of us that we can develop a molecular disease along with it. Okay? This is how it goes, and this is how it can be treated. Now, let's move on. Our next one, a very important, which is known as fermentation processes. This is a process which is widely used in industries for production of many products. Okay, so let's understand that what are the fermentation processes and how we can obtain it. Okay. Slow decomposition. The slow decomposition of organic compounds is called fermentation. Basically, it's a degradation, slow degradation of any organic compound is called fermentation. Fine? This is the principle behind souring of the milk, better purification of meat, and preparation of wine and vinegar. This is basically used in preparation of alcohol. Right? Fermentation is the most important thing for us in our day-to-day life also. Simple example we can take is curd. We can have a batter. South Indian recipes are generally made up of a batter which is fermented in nature. Right? Actually, fermentation increases its nutritional values. It increases the amount of vitamin B12 which is present in it. Right? So we can improve it with betterment of that particular fermentation process. Right? We have seen that pickles from generations and generations, we create pickles. This particular pickles are basically the fermented ones. The ones which are stored in the oil or in sugar, in very high amount of sugar. And it gives rise to that smooth and more nutritional values are developed into it. Right? Now, let's take an example of it. So, fermentation was the earliest method used for preparing alcohol in industries. This is still used for the manufacture of alcohol and alcoholic drinks like beer, wine and rum, etc. Right? Almost each and every kind of alcohol is produced through fermentation process only. Right? Now, let's take the examples of all these things here. So the first is production of ethanol and acetic acid. Right? Production of ethanol and acetic acid is generally possible through and through fermentation process only. For that we can use different resources. But here we are going to use molasses. Okay, we will have here an example of molasses. That from molasses, how we can produce ethanol. Okay, let's take a look on this kind of processes step by step, because these processes are actually step by step processes. Fine. First, the syrup left after the separation of cane sugar or beet sugar crystals from the concentrated sugar cane juice is called molasses. Okay. After that, or it is a dark colored syrupy mass and contains about 30% of uncrystallizable sucrose and about 32% of invert sugar, which is a mixture of glucose and fructose. Right? Now the different steps which are involved in the manufacture of ethanol through fermentation of molasses are: number 1, dilution. Okay, the molasses is diluted first with water until a concentration of 8 to 10% sugar is obtained in a solution. To discourage the bacterial growth, this is acidified with little sulfuric acid, and if sufficient yeast, basically food for the ferment, is not present, a nutritive solution of ammonium salts is added into it. Okay, this is the first step, which is known as dilution. Now, we will move ahead to the next one, which is known as alcoholic fermentation. So, the fermentation sets in. Now, the fermentation sets in, and the enzyme, which is known as organic catalyst, invertase present in yeast, converts the sucrose into glucose and fructose. Okay? Xylase, which is another enzyme which is present in yeast, converts the glucose and fructose into ethanol and carbon dioxide. Now, the carbon dioxide form is allowed to escape, but air is not allowed to enter. Okay? In presence of air, ethanol form will be oxidized to acetic acid. How it is possible? We will observe it through the example. Right? Here, if you can see, C12H22O11 plus H2O. We mix it together in the presence of invertase and in yeast. We will have a glucose and fructose, that is C6H12O6 plus C6H12O6 are produced. Now what will happen? In the presence of xylase enzyme, in the presence of yeast, that glucose or fructose gets converted to ethanol, that is C2H5OH, and CO2 is produced. Right? We just have to remove the CO2. We don't have to add the air into it. Okay, in this manner, ethanol will be produced. Fine. Now the next step will be distillation process. Right? Such kind of fermentation is generally completed within 3 days. Now the carbon dioxide obtained as a byproduct is recovered and can be sold. We have to just do the fermented liquor, which contains about 9 to 10% of ethanol, and is called wash or what? Is distilled in coffee still, which is distillation of wash in coffee still, to remove the water and other impurities are present in wash. Just remove the impurities and sell them. Right? This is just only 3 days process, and we can produce it within 3 days. Right? Last but not the least is rectification. Right? In rectification, what we will do? The alcohol obtained in such a manner which contains other impurities besides water. Right? These impurities are further removed by fractional distillation. Low boiling impurities like acetaldehyde distill over as the first distillate or first fraction. Okay. The middle fraction contains about 93 to 95% of alcohol and is called rectified spirit. That's why we know this particular process as rectification. Okay. Often distillation and rectification is carried out in the same operation. Okay. These both are generally done in the same operation, and we get our rectified spirit in that one only. Okay. This is the thing, this is the process with which we can obtain ethanol from the fermentation of molasses. Right? Generally, it will done within a span of 3 to 5 days. A proper ethanol can be obtained within this particular time frame. This is all for this session.